we going? One second, let me yeah, there we go. Oops, there we have it. Okay, well, thanks for tuning in. And as I said yesterday, I'm going to be going over this review called The Testis in Immune Privilege. So one of the things that I'd like to mention is there are areas in your body that your immune system is suppressed in order to help protect those delicate tissues from the immune system and from autoimmune disorders. So, for example, there is the eyes would be an example of an immune privileged site. The testes would be an example of an immune privileged site. And before I go into it, I will just do a quick Wikipedia of what immune privilege is so that everyone's on the same page. So here are the examples: the eyes, the placenta, and the fetus. So if you think about this, the placenta and the fetus have foreign DNA, they have foreign proteins, and so you don't want the immune system of the mother to damage the tissues of the placenta and the fetus. So that's why they're one of the immune privileged locations, and that could present problems for certain diseases, because if some diseases are able to cross through the placenta and cross into the fetus, then it is possible that there can be more, what would the word be? There can be more complications with that pregnancy. Continuing, the testicles, the central nervous system, and the anagen hair follicles. So, yeah, so your hair follicles can also be an immune privileged site as well. Now that I think about that, I wonder if that's why acne can be such a big problem. I'll look into that as well. Regardless, so here we'll go over the summary, and this is a long review, so I don't know if I'm going to do two videos or not, but either way, we'll have a lot of face-to-face time, and by face-to-face I mean my face, my mug, and you webcam. So let's go. Summary. The production, differentiation, and presence of male gametes represent inimitable challenges. Okay, wait, 1 second. I actually don't know what that word means. So, so good or unusual as to be impossible to copy. Oh, okay. Okay. You know what? You learn. I'm learning language when I go over these as well. That was definitely not on the GRE. But inimitable challenges to the immune system, or challenges that cannot be seen in any other places. As they are unique to the body and appear long after the maturation of the immune system and formation of systemic self-tolerance. So, so here, how do I phrase that? So your immune system will basically kill all of the, as the immune system matures, you end up killing a lot of the immune cells that end up being self-reactive. You kill the immune cells that create antibodies that end up damaging your own tissues. I forget exactly what the name of the process is, but basically that is a way to prevent you as a organism for having white blood cells that are presenting antibodies against certain antigens. How do I phrase that? Basically, there are other white blood cells that are presenting antigens on them, and they're presenting antigens that are coming from your own genetic material. And what happens when a white blood cell matures is that white blood cell will basically come into contact with a bunch of these antigen presenting white blood cells. And if it ends up reacting with any human protein, then that, unfortunately, that white blood cell ends up getting destroyed. And that way, that white blood cell doesn't go on to attack your own body later on. So going back to this sentence, the production, differentiation, and presence of male gametes represents an inimitable challenge to the immune system, as they are unique to the body and appear long after the maturation of the immune system. So you don't start producing sperm cells until you are hitting puberty. And I mean, ages range, right? So I mean, it's anywhere between, what is it, 9 and, no, not 9. That's a little early. I do hear that some people are hitting puberty earlier now, especially uh, there are worries about contaminants in water supplies that might initiate um, puberty earlier than expected. But I believe it's mostly for girls that are experiencing that. But let's just say late elementary, middle school is when they're hitting puberty, right? So by that point, you've already gone through the process of removing all these self-attacking antibodies or self-attacking white blood cells, which produce those antibodies, and therefore, as you are producing these new sperm cells and you're producing these proteins that are associated with the new sperm cells, those are not antigens that your body has screened for already. So this presents a big problem for your immune system because if your immune system's attacking your sperm cells, well, you're going to be infertile. But there are ways to get around it, such as immune privilege. So we will continue. Known to protect germ cells and foreign tissue grafts from autoimmune attack, the immune privilege of the testis was originally and somewhat simplistically attributed to the existence of the blood-testis barrier. So you might have heard of things like the blood-brain barrier, where it is very difficult for large molecules to go from your circulation system or your circulatory system into the central nervous system fluids. So that would be the blood-brain barrier, but there's also a similar barrier between the blood and the reproductive tissues of the testes. And so the previous model was that due to this tight control between the circulatory system and the testes tissue, we assumed that that was playing a major role in creating this immune privilege and protecting the sperm. However, recent research has shown a previously unknown level of complexity with a multitude of factors, both physical and immunological, necessary for the establishment and maintenance of the immunotolerance in the testes. So, besides the blood testes barrier and diminished capacity of the large testicular resident macrophage population to mount an inflammatory response, it is the constitutive expression of anti-inflammatory cytokines in the testes by immune and particularly somatic cells. So a lot of people like to talk about the cytokine storm or the cytokines, which are hormones that are released, if I remember correctly, predominantly from white blood cells or immune cells in order to help the body attack infection. Well, a lot of them are pro-inflammatory and can cause a lot of symptomology, but some cytokines, according to this, are anti-inflammatory as well. I wouldn't be able to tell you which cytokines are anti-inflammatory, just know that they are a different subset of the cytokines. Anyways, it is constitutive expression of anti-inflammatory cytokines in the testis by immune and particularly somatic cells that represent an essential element of local immunosuppression. So I should also say it is constitutive. It is always happening. It is always happening that the body, or the testes, are producing these anti-inflammatory cytokines. Let me look at this real quick. Large testicular resident macrophage to mat. So yeah, I don't know if it's from macrophages that are producing these cytokines, but they are constantly releasing these hormones in your testes, which prevent your immune system from mounting a response within them. Okay. The role of androgens in testicular immune regulation has long been underestimated, yet accumulating evidence now shows that they orchestrate the inhibition of pro-inflammatory cytokine expression and shift cytokine balance toward tolerogenic environments. So tolerogen, think of generating tolerance. So it's generating tolerance in this environment to whatever antigens are presented. Furthermore, the role of testicular dendritic cells in suppressing antigen-specific immunity and T lymphocyte activation is discussed. Finally, the active role mast cells play in and mast cells are another type of immune cell, play in the induction and amplification of immune responses, both in infertile humans and in experimental models, highlights the importance of preventing mast cell activation to maintain the immune privileged status of the testes. So, and I need to further mention that this is not my field at all. I'm just going through this and trying to make sense of it and trying to, for lack of a better word, regurgitate it for y'all so that you're also able to follow along with me. So if I do make mistakes and if I'm not able to fully represent what the author is trying to intend, you can leave that down in the comments. That way I can correct it for a following video. Introduction. So at the time of puberty, after the establishment of immune competence, male germ cells enter meiosis, beginning their complex transition into highly specialized spermatozoa. So the germ cells are the, if someone said, think of germline, think of, these are the cells that have the full chromosome number of the living organism, and what happens is this germ cell will split twice in a method which has the individual spermatozoa have half of the chromosomes that the normal organism would have, and therefore this sperm is going to be containing half of the genetic material of the original organism. But this happens during puberty. During the process, a myriad of surface and intracellular proteins is expressed, yet these new autoantigens are tolerated by the testes. So these are new proteins that have never been expressed before on the surfaces of these new cells, and since they have never been expressed before, we would assume that your immune system would recognize them and then start to attack them, right? However, the immunogenicity of the proteins is not diminished as shown by their ability to induce strong autoimmune reactions when injected elsewhere in the body. So if you take sperm, your own sperm, and you inject the sperm into a different location on your body that is not the testes, and we're going to assume that the injection is not going to be into an immuno-incompetent area, or not into an immunoprivileged area, you will mount a response against it. So you will actually have an immunological or an allergic type of reaction against your own sperm if it's injected into your body, right? So don't go testing that out, please, but just so you know, you will show an immune response to your own sperm. So it's not as if the immune system has learned to tolerate your sperm. It is simply being suppressed in the locations that the sperm is at. Okay, so rather, it is the testis itself that confers protection. Initial suggestions that the testis was an immune privileged site was substantiated experimentally with histo-incompatible allo and xenografts placed into the interstitial space of the rat testis, survived and prospered for indefinite periods of time. So what they're saying here is they took histo-incompatible or xenografts. So histo means like antibodies, so an antibody incompatible. Allo, which I would believe is from the same species. Let me look it up. Allograft. And if you look this up, you'll realize, okay, allograft. Let's go. Yeah, I'm not biology. Yes, so from one individual to another of the same species. So yeah, I was correct in that. So an allograft would be tissue that is coming from another member of your species, but remember it's histoincompatible. So this is coming from this tissue from the other species, even though it's still your own species, is still not your own tissue, and therefore you can mount an immune response, and that's what they mean by histoincompatible. So, for instance, this is why when it comes to organ donation, it's important to find good matches for organ donation. That way, your immune system doesn't mount an extreme response to it. But here, they're saying if we take histoincompatible tissue from either the same organism or a different species of organism, and if we put it into the interstitial space of the testes or just into a location of the testes, the rats not only tolerated it, they survived for indefinitely. So the immune system was not mounting a response against these foreign tissues in the body um, because they were put into the testes, right? So similarly, ectopically transplanted allogenic Sertoli cells not only survive, but also when co-transplanted with allogenic pancreatic islets, resistant rejection without additional systemic immunosuppression in animals. More, gosh, that was a mouthful. Let me reread that again with the right pronunciation. So similarly, ectopically transplanted allogenic Sertoli cells not only survive, but also when co-transplanted with allogenic pancreatic islets, they resist rejection without additional systemic immunosuppression in animals. So this immunosuppression or additional systemic immunosuppression, oftentimes when we do transplants, we need to put the host or the recipient of that transplant on some form of immunosuppression. And the reason is we don't want the person's immune system to attack that tissue, especially if they need it. So here they're saying if we take the Sertoli cells, which are a type of uh, what's it called, a testicular tissue. So I'm gonna search that just so that everyone has it. Is they are the nurse cells of the testicles, and it's part of the seminiferous tubule, and it helps in the process of spermatogenesis. So we will go over this later. There will be some photos within the paper, but just so you know, that is what a Sertoli cell is. So if we take the Sertoli cell, they can survive it when they're co-transplanted, um, and they can also survive when they are co-transplanted with allogenic pancreatic islets, which is another type of cell type from the pancreas. So this is where insulin is produced, if I remember correctly. So let me just do a quick search on that just to make sure I'm not lying to all of you. They are a group of cells within the pancreas that release hormones. Um, so it's multiple hormones, but insulin is one of the hormones that the islet cells uh, release. Continuing, and they resist rejection. So even though you would presume that you would mount an immune response against this, because it is located within the testes, they are able to survive and exist without immunosuppression. Anyways, more recently, the transplantation of spermatogonia into germ cell depleted testes could restore spermatogenesis, even across species, borders in some instances. So this is interesting. I think I remember. I think there was a Batman cartoon that went over this concept before. But basically, if you infertilize an organism, if you remove their germ cells um, from their testes, they're infertile, right? they're no longer producing sperm cells because sperm cells come from the germ cells. So the germ cells are nursed by the Sertoli cells into the. If I'm remembering this correctly, I might be my memory might be refreshed later in this, but they're nursed to maturation in with the Sertoli cells into the mature sperm cells, right? So if you get rid of all the germ cells, you have no sperm cells. But if you take germ cells from sometimes from a different species and you inject those germ cells into the testes of an infertile host organism, you would normally think that the immune system would kill it. You would kill the foreign germ cells. But rather than doing that, they live and they exist in the they often will survive within the testes of this other organism, and the sperm will be produced. So it's not just they're surviving; they are actually re-emulating the entire process in the new host organism, which is quite remarkable. As transplantation of tissue fragments occurs in the interstitial space and spermatogonia are injected into the lumen of the rete testes, both compartments of the testes are permissive for allo and xenoantigens. Let me just read that. They're just saying that this location is permissive for having these antigens be released. Yeah, I think that's what it is. So there is general agreement that immune privilege is an evolutionary adaptation to protect vulnerable tissues with limited capacity for regeneration. So this is what we were, therefore avoiding loss of function. So this is what I was saying before, is that the immune system is very caustic. The immune system is very damaging. So we, especially for cells that do not recover easily, so such as your nerve cells or your the cells within your eyes or your testicular cells, since they do not recover easily, it is important to protect those locations from the immune system itself. And that is the presumed model right now. For the testes, this protection means safeguarding reproductive capability. While protection of the developing germ cells from autoimmune reactions under normal conditions is evident in all species, there are distinct interspecies differences, as shown by transplantation experiments, where primate testes failed to sustain grafts of monkey thyroid or mouse testes as a recipient of human testicular xenografts. So there, within the organism, this is definitely happening no matter what organism we're talking about. But whatever mechanisms are being used within that organism might not necessarily cross into a different organism. So what we're seeing, presumably, would be, I wouldn't call it I wouldn't call it not convergent evolution. Was it? Yeah, it wouldn't be convergent evolution. It would just mean that there are probably multiple strategies that are being employed for each species, and it is not irreducibly complex. So there are some instances where you could probably add or remove certain protective strategies, and it still works within the same species, but it might not be compatible in a different species if you do a transplantation. Anyways, notwithstanding its immune privilege status, the testis is clearly capable of mounting normal inflammatory responses, as proven by its effective response to viral and bacterial infections. So you can have your testes swollen if you contract certain diseases. So this is something that one of the reasons why I brought this up, because I believe the term is orchitis, and it is when the immune system ends up inflaming the testes, And this happens with quite a few coronaviruses across species. So if I remember correctly, with MERS and SARS-1, testicular inflammation was an actual issue with those, and presumably it's because often the testicular tissues are expressing the the receptors for these viruses. Now, granted, it's not just the receptors you need; you also need high levels of specific proteases to allow the viruses to enter the cells. And then also with the testes, they they do a good job at protecting that testes blood barrier is very good at protecting the testes from viral and bacterial infection. However, like everything in biology, it's a very noisy field, right? So nothing is 100% perfect, and so in the cases where that failsafe fails, then you can have, like they say here, notwithstanding, the testis is clearly capable of mounting normal inflammatory responses. So yeah, so it happens. Some of those are STDs, so you gotta, if you choose to be sexually active, you need to, especially, well, no matter what sex you are, you need to be tested regularly if you are partaking in uh, more liberal forms of sexual expression. Continuing, in in pathological circumstances, the misbalance between the tolerogenic and the efferent limb of the testicular immune response can lead to the formation of autosperm antibodies, and in rare instances, epididymoorchitis in humans. Okay, a lot to unpack here. This is why science is hardly accessible, right? They're using so much jargon while they're writing this. So the misbalance between the tolerogenic and the efferent limb. So let's look up what that means, because I don't even know what the efferent limb. So Ru-NY, and serves as the primary recipient of food after the surgery, while the um I'm not sure if this is the right thing that I'm wanting to look up. So used to reconstruct the gastrointestinal tract. Oop, okay, did not mean to show that. It's between the tolerogenic and the, yeah, this is definitely a completely different thing than what Wikipedia is bringing up. But what I'm getting from this is that there is a push and pull between wanting to tolerate everything within the testes and the afferent limb, which I'm going to ascribe as the opposite of the tolerating aspect. And if there's a misbalance within that, it can lead to the formation of autosperm antibodies, so the production of antibodies against your own sperm, which I don't know about you, but that doesn't sound pleasant, especially in rare cases, this epididymoorchitis in humans. So the epididymis is um, you have your testicle, and the epididymis is one of the tubes that is sort of connected to the testicle where a lot of the seminiferous tubes go through, if I remember this correctly. So let's look up epididymoorchitis so that we can get a better uh, look into this. So, and as always, I'm gonna do Wikipedia because that is, so it might just be another more complicated way to say epididymitis, which is the inflammation of the epididymis. Um, we'll drink to this. So just so you know, with epididymitis, which is inflammation of the epididymis, um, yeah, and it's the curved structure at the back of the testicles. So you have your testicles. It is the tubes that are coming off of the back of it, which, and then I believe those tubes, which are still connected to the testes, ends up coming off to go to the what's it called, the prostate, if I remember correctly. Um, anyways, characterized by inflammation of the epididymitis, a curved structure at the back of the testicle. Onset of pain is typically over a day or two. And I'm just going to look up orchitis right now because that might be better for y'all. So it's inflammation of the testes, and it can also involve swelling, pains, and frequent infection, particularly of the epididymitis or epididymis, as in epididymitis. The term is from the ancient Greek orchis. Yeah, same root as, oh really, same root as orchid, really. The more you know. Okay, well, we're learning a lot. Just found out that testes inflammation comes from the same root word as orchid, but you know, makes sense. Not at all, but anyways, immune infertility is now estimated to be a considerable cause of childlessness in couples seeking medical assistance. Over the years, numerous studies have highlighted the impact of the immunological response to spermatozoa, which are one of the not yet completely matured but close to matured sperm, in the form of anti-sperm antibodies on fertility. Acting, let me just make sure I have that right as well. Oh, maybe nope. I was wrong. So it is the absolutely mature motile sex cell of animals. So it is explicitly when it is completely mature. So anyways, in the form of anti-sperm antibodies or ASAs, and they refer to ASA multiple times in this paper on fertility. So acting on multiple levels, ASA significantly impairs sperm fertilizing capacity. For example, by affecting sperm motility, the acrosome reaction, penetration of the cervical mucus, binding to the zoning pellucida, I don't know how to pronounce it, and sperm oocyte fusion. So these are all, so sperm motility, as you probably are aware, the sperm have the head and their tail, and the tail flagellates back and forth. But if you are making antibodies against proteins on that tail, that could seriously hamper its ability to flagellate around, which would hamper sperm motility. So that would be one way to become infertile. The acrosome reaction. So on the head of the sperm, you have the acrosome, which is a just another structure that has a bunch of I don't know how caustic they are, but it has chemicals which are important in helping the sperm get into the cell or get into the egg. So what I'll do is I will search Google so that we can see this again. So as you can see, the sperm is trying to get into the egg, right? And so the acrosome reaction is happening right here, where this acrosome right here, and here you can see this is called the acrosome granule. This has enzymes that allow the cell to start, for lack of a better term, burn through the protective coating of the egg, right? So here we will, during fertilization, a sperm must first fuse with the plasma membrane and then penetrate the female egg cell in order to fertilize it. Fusing to the egg cell usually causes little problem, whereas penetrating through the egg's hard shell or extracellular matrix can present more of a problem to the sperm. Therefore, sperm cells go through a process known as acrosome reaction, which is the reaction that occurs in the acrosome of the sperm as it approaches the egg. The acrosome is a cap-like structure over the anterior half of the sperm's head. So, yeah, this is it. So that is that. And then penetration of cervical mucus. So the cervix has uh, it it finds ways to be selective in which things are able to penetrate into it because uh, it is somewhat exposed to more pathogens and more microorganisms than most other parts in the human body. So you need to have protective measures put in place to prevent infection, right? So the cervical mucus is one of those barriers that help prevent that from happening. So by the sperm cells, therefore, have strategies to get through this cervical mucus. But if you have antibodies that are produced against the sperm, that could inhibit the sperm's ability to penetrate through the cervical mucus. So let's do I'm gonna go to Wikipedia. Okay, so it'll just be here, but mucus. Cervical, should I go through this? Such as the creating model and buildings due to I'll just go to cervical mucus. That way we can better understand it. So several hundred glands in the endocervix produce 20 to 60 milligrams of cervical mucus a day, increasing to 600 milligrams uh, daily, 600 milligrams of daily mucus around the time of ovulation. It is viscous because it contains large proteins known as mucins. The viscosity and water content varies during the menstrual cycle. The mucus is composed of around 93% water, reaching 98% at mid-cycle. These changes allow it to function either as a barrier or a transport medium to spermatozoa. So I should amend my first statement. It's not just a barrier. It is also able to help transport. It's a transport medium for the sperm to get to the egg. And the one would presume that the healthier the sperm or the more fertile the man who the sperm came from, that their sperm has better strategies of rather than of when it runs into the mucus, does the mucus serve as a barrier or is it a transport medium? So the spermatozoa have to figure out ways to do that. And so the more fertile man would presumably have better strategies to utilize the mucus as a transport medium versus a barrier. It contains a bunch of stuff. It contains amylase, really. I really. So for those of you who don't know, amylase is an enzyme that breaks down starches. So amylose is in like potatoes, breads. So there's amylose and amylopectin, and those are polymers of glucose. And amylase is there are multiple amylases, but amylases generally break down carbohydrates into simpler, smaller sugars, which um, them get digested by different enzymes, like glucoamylases or other enzymes that can break those simple sugars into glucose, and then those glucoses can be used for fermentation and the like. So that is interesting that there are amylases found within the mucus of the cervix. Yeah, I wouldn't know what this is for, but you know what? I'm kind of curious, so I'll probably look that up. So Google is gonna learn a lot about my brain today. Continuing. So that is penetration of the cervical mucus, and then binding to the zona pellucida, which is just another one of the layers around the egg and the sperm oocyte fusion. Um, so obviously the sperm has to fuse with the egg to do the fertilization. And if antibodies are being generated in any step of this process, those antibodies can interfere with the proteins on the surfaces of the sperm to do their function, which would um, therefore stop the sperm from being fertile. It would, and then it would also stop the man who produced the sperm from being as fertile as a result. Anyways, antibodies directed to sperm antigens can be detected in seminal fluid and seminal plasma in men, as well as in cervical mucus, oviductal fluid, or follicular fluid in women. They also occur in blood, serum in men and women, but these appear to be iso anti-sperm antibodies, which are not important for fertilization. Let me read this again. They occur in the blood serum of men and women, but these appear to be iso anti-sperm. Let's look up iso antibodies just to. So the how do I phrase that? They also occur in blood serum of men and women, but these appear to be iso anti-sperm antibodies. And what this is reading to me that these aren't necessarily the antibodies against your own sperm, so it's not an autoimmune antibody. It's against sperms in general or other sperms of the same species. Which I was thrown off because it said uh, blood serum of men and women, but women don't. Bi- Biological women don't produce sperm, so I was trying to understand why they even they mentioned that in the same sentence. But anyways, they're saying this is not important for fertilization because they're dealing with sperm of a different person. Only the antibodies that are bound to the sperm are considered to be of real significance for fertility. In 5 to 12% of infertile male partners, anti-sperm antibodies is found in the seminal plasma or attached to the surface of spermatozoa. And this is not good. Now, what I would like to know, just to be thorough, is what percentage of fertile men have anti-sperm antibodies in the seminal plasma or attached to the surface of spermatozoa. That would tell a lot, because if in fertile men it's also 5 to 12%, then that's not saying anything. But here, oh wait, in contrast, autosperm antibodies were also detected in the semen of or blood in men of proven normal fertility. So I wish that they would have left that in. I wish they would have given me the percentages for the normal men, but they decided not to do that. So maybe, yeah, I don't know. In contrast to anti-sperm antibodies, autoimmune responses against the developing germ cells within the human testis has not been studied very extensively. The most commonly used model for the investigation of autoimmune-based inflammation, testicular impairment is experimental autoimmune orchitis, or EAO. So remember the orchitis, apparently from the same Greek root word for orchid, um, is basically inflammation of the testes. And the experimental autoimmune orchitis means in a laboratory setting, you can create an autoimmune response to cause the inflammation of the testes, and that is called EAO. Um, and in this case, a rodent model based on active immunization with testicular homogenate and adjuvants. So an adjuvant is something that they put in, say, vaccines or things that they inject into you, which activate your immune system, and they make your immune system much more likely to generate a strong response at that injection site. So, if I remember correctly, aluminum oxide is a very common adjuvant because you will often form a strong response against that. So, um, yeah, they say it's to help the immune system in response to a vaccine. Um, Mechanism as stable. I'm not going to get into this, but I just want y'all to know that's what it is. So, and then testicular homogenate. So, when I was young, I used to buy. I didn't used to buy. I used to drink a lot of milk, and oftentimes the milk had a sticker on the cap that said "homo" on it, which is short for homogenization. Because when you homogenize things, you are basically blending it or putting things through a process in which the particles are evenly distributed throughout the material, or homogenized, if you will. And the the reason for homogenizing milk is because the fat in the milk will easily rise to the surface or rise to the top of the milk. And a lot of people don't want to have a chunk of cream on the top of their milk. So the homogenization process is something that can take those fat globules and make them small enough so that they stay suspended in the liquid or in the bulk of the liquid for far longer uh, than the large globules of fat, because the large globules of fat, since they're much less dense, are able to rise faster because there's a greater buoyant force on those fat globules than the smaller ones. Anyways, at least on a per weight basis. Anyways, what they did with these mice, and it's kind of gross if you think about it, is they took the testicular homogenate, so they took testes and they blended it up, and then they added an adjuvant, which is something that makes the immune system very active, and then they injected that into these rodent models. And then when that injection happened, then you get this experimental autoimmune orchitis, or you can see the inflammation of the testicles of the mice when you give them an injection of testicles and an immune activator, which would then tell the immune system this is a foreign invader and I definitely need to attack it. Well, crap. Well, after you do that, then it ends up screwing up the rat's own testicular tissues. And then there's a lot to be said about that be a form of mimicry, right? Where, um, for instance, some bacteria and some viruses have found strategies to mimic the proteins of their host or their victim organisms. And because they have learned to mimic these proteins, when the when you or when the infected creature ends up producing antibodies against the pathogen, those same antibodies can often cross-react or attack their own body as well. And this is a strategy for the viruses and the bacteria, because if the antibodies are being used to attack their own body, then they are no longer effective at attacking the virus. So you have to make a lot more antibodies to make up for the fact that some of them are used against the body of the host, right? Anyways, continuing, this experimental autoimmune orchitis can be also adoptively transferred into syngeneic recipients by CD4 plus T cells or testis-specific T cell lines. So uh, if you take these white blood cells that you know that attack the testes proteins, then and if you take them and you inject those immune cells into a naive rodent or a naive model, those immune cells will continue to live within the rat. But then they start attacking the testes of the rat when they're in the rat. So this tells you that you can, with just the immune system, with nothing else, with just the immune system, you can cause inflammation of the rat testicles. You can cause this experimental autoimmune orchitis. Continuing, whereas depletion of the CD4 positive T cells in vivo or in the living organism inhibits the disease. So a if you take a organism that you've created this experimental autoimmune orchitis and you kill all of their CD4 positive T cells, what that happens is that cures the poor creature of the testicular inflammation. Uh, it's actually really kind of sad, and I do sympathize with animal rights activists that would argue that this is pretty. It's pretty sad. I would hate to be this mouse because you either are in the position of having inflamed balls or you lose your immune system. And either of those are not very pleasant lives to live. Anyways, the clinical term orchitis is particularly attributed to acute symptomatic disease. So we tend to reserve orchitis just for these acute uh, inflammation of the testicular tissues. Uh, and we might have a different word if it's a more chronic or long-term type of disease due to local or systemic infection. Whereas subacute or chronic asymptomatic inflammation of the testis, including non-infectious disease, is difficult to diagnose and therefore likely to be ignored. So this is one of the biases in the sciences that, uh, especially the medical sciences, that need to be addressed, is that if someone always has their testes inflamed, how are they supposed to know that that they're sick? Right? It's sort of like a lot of people who have gluten intolerance and have autoimmune diseases that are caused by consuming wheat proteins, which is a problem where certain plants cause autoimmune reactions because the immune system has mounted a response to those antigens for no reason really, but unfortunately it's happened, so they have to cut that out of their diet. Where was I go? Oh yeah, so those people, the people who have this ongoing inflammation at all periods of times, how are they supposed to know that they're unhealthy, right? How are they supposed to, if your baseline is different from someone else, how are you supposed to like realize, oh, I'm actually sick, I'm actually wrong? I had a buddy who had a really bad gallbladder, right? And it just did not produce the it was either the enzymes or the bile acids or the bile salts to properly digest food. And he basically um, had been miserable for a very long period of time. But if that's your normal, right? And then it was only after he had to go to the hospital and they said, "You've been pretty miserable for quite some time, haven't you?" And yeah, he said, "Yeah, I've actually been pretty miserable for quite some time." And then they ended up removing the gallbladder and put him on a special diet. And then that changed everything. That changes everything. When you finally realize, oh yeah, I actually was oppressed by my own biology, right? So here, this is one of the biases in the medical terminology where a lot of chronic issues might not necessarily be properly diagnosed or studied because they are chronic and people don't know to recognize it, and people aren't going in for general checkups to provide this information for the medical community, and by extension, the world community, because data is everything. Data is able to uh, elucidate a lot of these problems. But if we don't even know what to look for, then we have imperfect sampling, if that makes sense. Continuing, the classification of rhinitis in humans depend on the etiology of the condition and can be caused by traumatic events as well as bacterial and viral infections. So this is what I was saying earlier about STDs. You gotta check yourself before you wreck yourself and others. Um, as in humans, it is usually associated with epididymitis, so inflammation of the epididymis, and this is painful. But it's not always caused by bacteria. It can be caused from other things. So, for instance, one of the things that can cause epididymitis is just having blue balls. So, this reminds me. There was there was a very big push for abstinence education for a lot of young people, high schoolers, and one of the things that I remember the one of these abstinence focused advocates spoke about was nothing's gonna fall off if you don't get off, or something along those lines. And I mean, turns out there are some serious health consequences that are the result of not regularly getting off. And one of those things that I should mention here is that with blue balls, I should also mention that oftentimes these abstinence advocates also advocated against pornography and against forms of self gratification, which is perfectly fine if you want to live that particular lifestyle. That is great. But there are serious health consequences that you might have to consider if you consent to that particular lifestyle. So if you are not getting off regularly, that could have some serious implications on chronic disease associated with your testicles. So, for instance, blue balls can cause swelling of the testes if you are particularly aroused for long periods of time. That can cause epididymitis, and periodic epididymitis can be associated, for one, with some of these autoimmune based inflammatory testicular impairments. So I mean, I want to watch what I say, because as I said, biology is noisy. Nothing is ever 100%. Nothing is ever completely set in stone. We have very large standard deviations in biology. I'm just wanting to warn my audience of 4 subscribers that uh, it is healthy to get off. It is healthy to regularly get off in order to prevent some chronic diseases, chronic non-pathogen diseases. So just be warned about that. And I'm not going to go too far into that. Regardless, I'm going to take a drink. Let me take another drink. Okay. Anyways, orchitis may also occur in conjunction with infections of the prostate and as manifestations of sexually transmitted diseases, such as gonorrhea or chlamydia, Trichomatis. Just call it chlamydia. Urethral pathogens, including E. coli, cause bacterial epididymolycitis. The most common cause of viral orchitis is mumps. Really? Okay. On balance, these data clearly indicate that the mechanism underlying immune privilege in the testis and its disruption by pathological alterations are matters of clinical importance and hence continued scientific interest. So, I mean, you could go without saying it. I feel like the reproductive organs are always going to be of continued scientific interest. Continuing, I'm an adult. Should act like it. The testis, as the male gonad, has fulfilled two major functions: the generation of gametes and the production of controlled release of sex steroid hormones, primarily androgens, with testosterone being the most predominant. The testis is compartmentalized histologically, so there are different compartments that have different like immune sensitivity and function, with androgen production and spermatogenesis being confined to distinct regions. So spermatogenesis takes place in the seminiferous tubules or germinal compartment, and androgen is synthesized in the Leydig cells or the Leydig cells in the interstitial compartment, which is interspersed between the tubules. In the human infant, true septa extend from the fibrous capsule, which surrounds the testis, and divide the organ into various lobules. In the adult human testis, the lobuli are present but less conspicuously, while in rodents they disappear completely. The geminal compartment of the testis is arranged within the highly coiled seminiferous tubules, which originate and terminate at the rete testis. Each tubule is surrounded by myoid pertubular tissue that provides structural support and contains contractile elements capable of generating peristaltic waves that transport the immotile testicular spermatozoa along the tubule. So, think peristalsis. It's what your esophagus does when you swallow food. So, the tubule is surrounded by myoid peritubular. So I believe this is like some form of muscle tissue. Let me look this up just to make sure. Yes, so it is smooth muscle cells that are surrounding these seminiferous tubules. And so these are sort of squeezing the tubules in order to move the spermatozoa before they have their tails made. They're moving them towards the prostate, or they're just moving them in general. And they also provide structural support. Continuing. Oh yeah, into the epididymitis. So it's going from within the testicles into that epididymitis, which is in the back part of the testicle. The peritubular cells do not form a tight junction barrier. Rather, they express a high number of cytokines, growth and differentiation factors, and together with Sertoli cells, secrete the components of the basal membrane, which encloses the contents of the seminiferous epithelium. The columnar Sertoli cells extend from the basal lamina towards the lumen of the tubules and constitute the main structural elements of the seminiferous epithelium. They are responsible for the physical support of the germ cells in addition to providing essential nutrients and growth factors. So here you can see these are the seminiferous tubules right here, and here's another seminiferous tubule, and the inner part of the tubule is called the lumen, And here you can see this is the PTC, which is the paratubular cells, which those are the smooth muscle cells that provide support and are doing the peristaltic movement. And here you can see these are the Sertoli cells, which are the nursing cells of these cute little germ cells that are maturing into a sperm cell. But while they're maturing, they're still not fully mobile, right? So these smooth muscle cells are squeezing these immature sperm cells towards the epididymitis. Okay, and here you can see that there are very tight junctions to help protect, or I should say, cause a separation between all of these immune cells in in the interstitial compartment, and separate these and their antibodies from the geminal compartment, which is within the seminiferous tubules. Right, so here the morphology of the testis. Sertoli cells traverse the whole of the tubules. Germ cells are in intimate contact with Sertoli cells at all stages of development, together with the surround. Oh, really? So yeah, maybe they even stay within these small areas as they're transferring through the tissue. There's a lot to remember. Like biology, like you can't. I will forget this by next week, just so y'all know. So, together with the surrounding myoid pertubular cells, or the PTC, they form the germinal compartment of the testis. The seminiferous epithelium, the blood-testis barrier, is maintained by tight junctions with neighboring Sertoli cells, dividing the seminiferous tubules into a basal and adluminal compartment. The interstitial space located between the tubules contains the Leydig cells, which are, like we said, those hormone producing cells. So these LCs, these are the hormone producing cells, and such as testosterone, and immune cells such as the macrophages. Macrophages, here's a macrophage, the dendritic cells, the DCs, and they're called that because of all these branches that are coming off of it. So dendrites, which is, I believe, Greek or Latin for branch. The mast cells, so these, and the T cells, as well as the blood vessels. So here you can see a blood vessel that is just running through this interstitial compartment. And this is obviously going to be providing nutrients and oxygen for the testes. And within the blood vessel, you will have the blood cells. And obviously, these aren't to scale, but they are providing it for you so that you can understand the process that's going on. This is fun. I like this. I think we should go for 2 hours. Anyways, mammalian spermatogenesis is a complex process of proliferation and maturation involving the interstitial or initial multiplication of progenitor cells by mitosis, which is then followed by the first meiotic division. The resulting cells, now called primary spermatocytes, divide to form secondary spermatocytes, and then divide again in the second meiotic division to form the haploid round spermatids. So these germ cells, so these progenitor cells, they are undergoing mitosis. They are remultiplying. They are existing, living their best lives, right? And then later, some of those progenitor cells will then move into meiosis rather than continuing on with mitosis. And in that process, they end up turning into the haploid sperm cells, right? Continuing, the successful transformation of the round spermatid into the complex structure of the spermatozoon uh, zone is called spermiogenesis and involves the removal of most of the spermatid cytoplasm in the form of the residual body, condensation of DNA in the sperm head, tail formation, and establishment of the acrosome, which is that thing that we said earlier. So as these mature, a lot of the DNA becomes much more compact, and a lot of the components within that cell have to be removed to make a much smaller sperm cell. A cap like Golgi-derived sac, which covers the nucleus opposite of the tail and releases lytic enzymes. So yeah, I was right by saying it's full of enzymes. It is full of enzymes that promote lysis, or they promote destruction of whatever's in front of it. And you need to have those enzymes in order to get into the egg. So all of the events in sperm production are highly coordinated within each region of the seminiferous epithelium and occur in a regulated cyclical manner that involves both endocrine and local, also known as autocrine and paracrine, control mechanisms. So remember, autocrine is when a cell produces a hormone, and that hormone ends up influencing its own its own cell, right? So you make a hormone, and then the hormone, uh, you react to it as your own cell. That's autocrine. And then paracrine is when you release a hormone, and then that hormone ends up, I yeah, it's still called a hormone, and it touches one of the cells nearby. That's paracrine. And then endocrine is when the hormones are made in a specialized tissue at a far distance in the body, and then it goes into some sort of circulatory system, and then travels across the body to its target cells, right? Anyways, the most prominent constituents of the interstitial space of the testis are the clusters of heterogeneous in respect to their physiological and structural features, androgen producing Leydig cells. So all of these Leydig cells, they said they're heterogeneous in that they have there's a wide variety of different structural and physiological features on them. So they're trying to emulate that uh, heterogeneity with these blobs over here. Uh, Anyways, possessing a microvasculature, the interstitium also contains macrophages, lymphocytes, and increasingly, with age, mast cells. Uh, these guys have recently described large fat fibroblastoid cells which compartmentalize the microvessels, the Leydig cells, and part of the seminiferous tubules. These cells, justifiably named co-cells, appear to produce extracellular matrix components such as decorin, fibroblast surface protein, and vimentin. So just these are different types of proteins that are secreted, and that's what they're called, extracellular matrix components, and are typically found only in the human testis. Interesting. The local variability in the extracellular matrix proteins is important in cell-cell interactions within the testis, particularly as some proteins are able to bind to various classes of growth factors, thereby forming a reservoir that is able to modulate the bioavailability of growth factors to the respective target cells. So let me read this just to make sure I fully understand it. So the local variability in the extracellular matrix proteins is important in cell-cell interaction within the testis, particularly as some proteins are able to bind to various classes of growth factors, thereby forming a reservoir that is able to modulate the bioavailability of growth factors to the respective target cells. So I think what they're saying here is that these compartmentalizations are able to prevent a lot of bleed through, and by preventing that, you can preferentially cause more paracrine and autocrine types of cellular signaling and you can control where the growth factors are at. I mean, I'm spitballing right now, so I don't know if that is exactly what's going on, but yeah, they're just saying that this compartmentalization is important. So I will trust their judgment. Anyways, the endocrine exit of the testis is the rete testis, a complicated network of intercommunicating slit-like channels which are lined by a flat or low columnar epithelium. The transition zone between the terminal segments of the seminiferous tubules and the rete has a specific arrangement of Sertoli-like cells, often forming a valve or a plug. The blood-testis barrier is terminated in this area. Subsequently, spermatozoa are no longer prevented from autoimmune attack, a status, a status confirmed by the observation that certain forms of autoimmune orchitis are first manifested in the rete testis. So they're saying here that in this transition zone, um, there's a special arrangement of these Sertoli-like cells that form a valve or a plug, but here the blood-testis barrier is it's not there anymore. So you can see more autoimmune attack of the sperm happening in this area. However, it should also go to note that if orchitis or the inflammation is caused in the testes at all, it will often first show itself in this in the rete testis. Interesting. Anyways, spermatozoa released from the seminiferous epithelium are immotile and passively transported through testicular fluid secreted by the Sertoli cells through the rete testis into the epididymitis. Spermatozoa mature during transit through the epididymitis, which consists of a single, highly convoluted duct, and finally gain fertilizing capability. The blood epididymitis barrier is different from the blood testis barrier and shows spatial differences and an age-dependent decrease in restricting permeability of molecules. So as you get older, the barrier between the blood and the epididymitis gets weaker and weaker. So this could have some serious implications on fertility in the future. In contrast to the seminiferous epithelium, T lymphocytes and macrophages are frequently found within the epididymal epithelium and in the lumen of the epididymal duct, pointing to an immune environment that operates differently from that of the testis. Yeah, so let me just scroll through. Let's see where a good spot to end is. I think we should do 2 more pages and then we'll finish for the first video. Anyways, the existence of a blood testis barrier was originally suspected when early studies found that certain dyes were excluded from cells inside the seminiferous epithelium. Later, ultrastructure of biochemical data confirmed and characterized this boundary to be highly specialized tight junctions, or zonula occludens, between neighboring Sertoli cells capable of restricting the passage of larger hydrophilic molecules, particularly proteins, through the intercellular spaces. So that was these right here. So the hydrophilic, so the water-loving molecules, and proteins, the surfaces of proteins are often more hydrophilic than they are hydrophobic. These are incapable of, or I should say generally incapable of getting through these tight junctions over here. Anyways, this limited access together with secretory activity of the Sertoli cells ensures that the composition of tubular or luminal fluid differs significantly from that of the interstitial fluid surrounding the seminiferous epithelium and creates a unique nurturing environment for the developing meiotic and maturing post-meiotic germ cells. So we are, every one of us are coddled from our very beginnings because the the sperm are very they're sensitive. They're very sensitive to external factors. So we want to create this nurturing environment for them, and so that is why a lot of effort goes into protecting and nurturing germ cells. Anyways, this blood testis barrier comprises of various integral membrane proteins, which in turn contain a number of interesting components, such as junctional adhesion molecules, or JAMs. Biochemists love their acronyms. Claudins 1 and 11, with claudins 3 to 5 and claudins 7 to 8 also identified in the testis. I don't know what claudins are, but yeah, and occluded. So just know that these are just different proteins that are secreted. The blood testis barrier divides the seminiferous epithelium into two distinct compartments. The basal compartment carrying the spermatogonia, leptotene, and zygotine spermatocytes, and the adluminal compartment with meiotic, pachytene, and secondary spermatocytes, haploid spermatids, and spermatozoa, which are all completely engulfed by cytoplasmic protrusions of the Sertoli cells. The main task of the blood testis barrier is to protect the developing germ cells from the immune system. So let me read this again just to make sure, because there's a lot of words in there, right? They got the best words. Um, Haploid spermatids and the basal compartments comparing the spermatogonia. So let's see if that is contained within this photo. So here's the lumen. So what they're talking about might be the difference between this area that it is where the Sertoli cells are. So this would be the adlumen area, and this would be the other area, right? The basal compartment and the adluminal compartment with the. Oh, okay. So I sorry. This part that is protected by these protrusions, these cytoplasmic protrusions of the Sertoli cells, this would be the adluminal compartment. I tell you, I understand why no one understands biology. Hell. So they're saying these cells are in the adluminal compartment, and these other cells are in the basal compartment. And I assume the basal compartment is the this part, but we're going to have to keep on reading just to make sure. Meiotic and postmeiotic germ cells, including the spermatozoa, which are produced at 150 million per day in rats, express a large array of neoantigens, so new antigens. So these are things that your immune system hasn't seen before, right? So since they are neoantigens that first appear during puberty, which is long after the establishment of self-tolerance, so the process by which you kill all of the self-attacking antibodies, um, with the instigation of spermatogenesis, the blood testis barrier is concurrently established and immediately sequesters post-pubertal germ cells from the immune system. So, with the instigation of spermatogenesis, so the beginning of the production of sperm, sperm cells, mature sperm cells, the blood testis barrier is established in order to prevent the germ cells from being attacked by the immune system. It is completely functional, and its integrity is maintained by the time the first preleptinine spermatocyte moved through the junctional complex by coordinated opening and closing of the barrier. Interestingly, As jams play a crucial role in leukocyte transmigration, it is tempting to speculate that they perform a similar role in the testes by facilitating the transfer of the leptotene-zygotene early spermatocytes through serotoli cell tight junctions. So this is why. So the leukocytes are a type of immune cell that is able to squeeze itself through other epithelial cells. And apparently the jams play a role in which, remember, these jams are the what is it called? They had the acronym. They're a type of protein. The junctional adhesion molecules, right? So these are dealing with junctions. So the junctional adhesion molecules play a role in leukocyte transmigration. So it's tempting to speculate. There is a hypothesis that these jams are playing a role in helping these leptotene, zygotene, early spermatocytes through these cell junctions. So these cells might be using jams to help them squeeze through these tight junctions. This is not proven yet. So impairment of blood testis barrier integrity has been observed during inflammation, infection, and trauma, which ultimately results in germ cell loss. So do not allow your balls to become inflamed because this is usually resulting in germ cell loss, which your germ cells play an important role in your own fertility. So if you want to have kids, improve your chances as much as you can, right? Mechanistically, elevated levels of tumor necrosis factor alpha, or TNFA, or alpha, and transforming growth factor, TGF beta, found in systemic and local testicular inflammation, have been shown to perturb the assembly of tight junctions in cultured Sertoli cells, probably by downregulating occludin expression. So when we do an experiment on cultured Sertoli cells, so they're not in the testes, we have them growing on some sort of plate, right? If we give them higher levels, of tumor necrosis factor alpha and transforming growth factor beta, and these are also found in systemic and local testicular inflammation. So these are hormones that we often see in inflammation. If we give those same hormones to cultured Sertoli cells, um, they have been shown to perturb or I would say dysregulate the proper functioning of these tight junctions in Sertoli cells. So if you grow these Sertoli cells on a plate and you add these growth factors that are, or these factors that are associated with inflammation, these tight junctions do not form properly. And they presume it's due to the down regulation of occludin, but that is not, proven completely yet. Despite the junction's ability to isolate meiotic and postmeiotic germ cells from circulating antibodies and leukocytes, it is now accepted that the blood testis barrier alone does not account for all the manifestations of testicular immune privilege. So you as a, not necessarily you, but your kind, uh, as possessors of testes, are probably using or utilizing multiple strategies in order to protect your sperm. Because if you have just one strategy, um, the term would be irreducible complexity, right? So if you are only utilizing one strategy, and if that strategy fails, the system fails, right? But presumably a healthy surviving organism is utilizing multiple strategies for all of their cellular functions, all of their tissues, and that way when one of the mechanisms screws up, when one of the mechanisms, let's say there's a mutation or there's a disease or a toxin, um, having multiple strategies for the same function can create redundancy to help preserve the life or the function of the organism and help them reproduce, help them pass on their genetics, right? So, anyways, this proposition supported by the findings that germ cell autoantigens are present in the basal compartment in spermatogonia and early spermatocytes, which are not protected by the blood-testis barrier. Moreover, the blood-testis barrier is incomplete in the rete testis, a location where immense numbers of spermatozoa with newly adapted surface molecules traverse through the epididymitis, making it a particularly susceptible region for the development of autoimmune orchitis. So what they're saying here is it can't just be the barriers and the tight junctions, because there are locations in your body that don't have these tight junctions, right? So we would safely assume that the immune system is probably going to be able to attack the sperm cells in these less protected areas. So there has to be another strategy at play to be causing a protective effect on these sperm cells in these more dangerous locations, right? But because in these certain areas, since they have less mechanisms in place, less safeties in place, then they are the first to experience inflammation and experience pathology if those other mechanisms are also put out of whack. Anyways, histopathological observation of mice in which this experimentally acquired orchitis has been elicited by injection of a mixture of viable germ cells revealed infiltration of lymphocytes, first in the tunica albuginea. I don't know how to pronounce that, but I'll let you read it. Close to the rete testis and tubuli recti, then spreading to the interstitial space. Both locations are well outside of the blood-testis barrier. Furthermore, these people showed extended survival of allografts that were placed under the organ capsule in the testicular interstitium. So remember, the testicular interstitium is, I believe, this location. The testicular interstitial compartment, I'm assuming this is the interstitium, so this is the area that still has white blood cells, right? So they still have white blood cells in these areas. And these allografts, so tissue coming from the same species, but that should elicit an immune response, that were placed under the organ capsule in the testicular interstitium. Showed extended survival. So it showed that if they did that, there was no immune response or attack if these allografts are put into the testicular interstitium. However, there was local injury of the seminiferous epithelium caused by the routine procedure of fine needle biopsies in humans, has been found not to cause orchitis. So even with, let me read this again real quickly. They have run-on sentences and it's making me nervous. While local injury of seminiferous epithelium caused by the routine has been found not to cause orchitis. Okay, therefore, some other mechanisms besides physical separation must exist to maintain testicular immune privilege, which requests more robust protection of the tolerogenic environment of the testis. So they also said here that even poking this even poking the seminiferous epithelium did not cause orchitis. It did not cause the inflammation or the autoimmune inflammation. So there has to be other things involved that is creating a more robust protective experience for our poor developing sperm cells. So continuing, endocrine regulation of testicular function and immune privilege. So the endocrine regulation of the testis has been reviewed extensively. As such, only a brief update with emphasis on immune privilege is provided in this section. Testosterone, follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH, are the most important hormones controlling testicular function. FSH and LH are both secreted by the anterior part of the pituitary, so that is in your brain, and acts through specific G protein coupled receptors. So, G protein coupled receptors, just know that these are. These are receptors on your cells, and there are a lot of them. Actually, what I'll do is I'll just look up the Wikipedia page. I believe they have to do with guanidine cyclase, but well, this is fun. So I think, okay, let me just double check. So these are also known as 7-pass transmembrane domain receptors. So they're called this because they have 7. So if we could imagine that this is sitting inside of a cell membrane, right? So you have your fatty inner layer and the two polar layers of your cell membrane. It has seven transmembrane domain receptors. So they're also called the 7TM receptors, or heptahelical, because very frequently the transmembrane helices, or the transmembrane structures, utilize these alpha helices as their preferred transmembrane secondary structure of choice, I should say. So direct the molecules outside. Yeah, so these are receptors that are on your cell membrane, and they detect molecules on the outside, and then they find some way to transduce that signal to the inside of the cell. And they're coupling with G proteins. And if I remember correctly, I'll just go to G proteins. I don't want to go too far into biophysics here. Yes, so guanidine nucleotide binding proteins that act as a molecular switch inside of cells. So, yes, I was right. Let me just. So, these are proteins that are able to bind to guanosine diphosphate or guanosine triphosphate. So, I'm not going to get into the physics of that. Just know that that is the mechanism by which they're transducing the signal into the cell, right? So in the testis, follicle stimulating hormone targets those serotoli cells, or those nerve cells, exclusively, and stimulates their proliferation during the perinatal and or pubertal period. In the adult testis, follicle stimulating hormone has tropic functions regulating the synthesis of a large variety of metabolites, such as lactate, pyruvate, and transport proteins, transferrin, antigen binding proteins, growth factors, activins, inhibitins, and cytokines. Luteinizing hormone, in contrast, acts on the Leydig cells by binding to its cognate receptor and consequently stimulating androgen production. So LH binds with the Leydig cells, which, remember, those are the hormone-producing cells, and then the follicle-stimulating hormone will bind with the Sertoli cells to stimulate, because remember, these are nursing cells, so they're stimulating the production of a lot of these other chemicals such as lactate, pyruvate, transport proteins. So these supposedly, I shouldn't say supposedly, but presumably would be playing a role in the nurturing and the maturation of the sperm cells. Testosterone, the main androgen, is the crucial hormone for the initiation and maintenance of the spermatogenesis by mediating its function through the androgen receptor expressed in the Leydig cells, Sertoli cells, and pertubular cells. So let me just double check everything. So the Leydig cells are also producing the testosterone, if I remember correctly. So yeah, so and then they're also okay. So the latest cells are producing these androgen hormones, and these androgen hormones are doing multiple functions. They are hormones. They're endocrine hormones, right? So they're going out into the body, reacting with receptors all over the body. All these androgen receptors all over the body, causing facial hair, causing more muscle mass, growth, all that shit. Or crap. Mm, this is a family channel. And then but it's also the antigens that are being produced are also coming back to the latest cell, reacting with their own hormones and causing more changes that way. And it's also secreting into these other cells, such as the Sertoli cells and the peritubular cells. So, and remember, these are here are the Sertoli cells and here are the peritubular cells. And remember, testosterone is a steroid. It is a non-polar. It's a hydrophobic molecule. So these hydrophobic molecules are able to. They're not like the proteins. They're able to squeeze through these tight junctions. They're able to go into the cytoplasm of these cells very easily. So that is how these latest cells they get activated by the luteinizing hormone, and then this is causing them to secrete more testosterone, which is then affecting everything in your body, but especially these Sertoli cells and these I forget it peritubular cells. So interestingly, the germ cells themselves are devoid of these receptors. Oh, so these latest cells, these are producing testosterone, and they're playing a vastly important role in the maturation of these germ cells. But these germ cells themselves do not have the receptors for the androgens. So that I think that is pretty interesting. I wouldn't have suspected that, but interesting. Meaning that androgens must regulate spermatogenesis indirectly through the receptor in the testicular somatic cells. So somatic cells means cells of the body. So your body has the somatic cells and it has the germ cells, and that just so you know that is these are cells that are not germ cells, somatic cells. The Sertoli cells being the main target. Peritubular myoid cells synthesize a number of androgen-regulating factors, which can also modulate spermatogonial and Sertoli cell function. And there is now accumulating evidence that the peritubular cells cell-secreted cytokines such as the TGF-beta, macrophage chemoattractive proteins, and leukemia inhibitory factor also directly influence leukocytes in the interstitial space. Of note, intratesticular testosterone concentration in rats is far higher than that needed for the maintenance of functional spermatogenesis. So, this must be in contrast with humans, where in humans, if you have low testosterone, that can have a very significant impact on your fertility, right? But apparently, in mice, let me just get this right. In the testosterone concentrations in rats is far higher than needed for maintenance of functional spermatogenesis. So rats generally, and I really should be explicit here. So this is lab rats. There is probably a big significance in the physiology of lab rats versus wild type rats, because lab rats have undergone successive rounds uh, of breeding, which can seriously hamper or change what evolutionary strategies they decide to go by, especially since when it comes to the production of rats, the more, well, what would the word be? The rats that produce a lot more progeny are probably going to um, be preferentially bred, if that makes sense. So that could be something that artificially raises the testosterone level of lab rats in in contrast to wild type rats. But I mean, that's just a theory, but I've been reading more about how the differences in the breeding protocols should be taken into account before making judgments between lab rats and the general mammal population. Continuing, in fact, intratesticular testosterone can be reduced by 60% without any adverse effects on sperm production and would still be tenfold greater than serum values. Okay. So they're saying tenfold greater than serum values, so tenfold greater than the concentration found in the plasma. I assume that's the type of serum they're talking about. And in addition to the well-known anabolic and spermatogenic effects, a role for androgens in downregulating pro-inflammatory cytokines has now been shown in both experimental and clinical studies. So these hormones, these androgens, are helping lower these pro-inflammatory cytokines. So they're helping control the cytokine response. Okay, just need to think about that for a little bit. Okay, so I'll move on. I might have something to say about that later. And incubation of stimulated human monocytes, macrophages, and several non-immune cell types with testosterone resulted in the suppression of adhesion molecules and cytokines such as interleukin 1, IL-6, TNF alpha, and increased production of anti-inflammatory cytokines such as interleukin 10. Several reports mention improvements in human autoimmune diseases such as lupus and rheumatoid arthritis following testosterone replacement therapy and testosterone-dependent enhanced susceptibility to infection, which in some studies could be reversed by orchiectomy. Is that when they take out the testes? Okay, so testicle removal. So anyways, and testosterone-dependent enhanced susceptibility of infection, which in some studies could be reversed by removing the testicles. So what would this look like? So if you are expressing high levels of testosterone or other androgens, those are probably going to have an immunosuppressive effect on whatever creature is expressing high levels of testosterone. But remember, the immune system, it's not just your immune system's working good or it's working bad. Your immune system is always ebbing and flowing between what it needs to do based on infections, based on how it's damaging your own cells. And so there are some cases where if some people are very susceptible to diseases, it might be due to some testosterone related pathway. So they might have a mutation which makes their receptors more susceptible to testosterone. So specifically the receptors that are on these immune related cells, they might have a mutation which makes them much more susceptible. So if you're expressing any testosterone at all, it might suppress their function too much, which could theoretically cause an increase in viral infection or an increase in what would the word be? Yes, susceptibility to infections. So by removing the testicle or by removing sometimes one testicle, you are able to help stop those types of infections from being as commonplace. In hypogonadal men, so hypo meaning low, so hypogonadal men, administration of progesterone was positively associated with serum interleukin 6 and E-selectin concentrations, which I believe are just cytokines, whereas the levels of anti-inflammatory cytokines dropped. So the introduction of progesterone, which is more associated with being a feminine steroid, this is increasing the pro-inflammatory cytokines while decreasing the anti-inflammatory cytokines. So this could be one reason why women tend to experience autoimmune diseases significantly more than men, right? In contrast, testosterone supplementation decreased the interleukin 6 levels, which is one of the pro-inflammatory cytokines, and a direct connection between sex steroid levels and testicular immune privilege was shown by these people. When in transplantation studies, rats pre-treated with estrogen to suppress the Leydig cell testosterone production promptly rejected intratesticular allografts, in direct contrast to the reaction of their untreated cohorts. So remember, we were talking about taking tissues from another member of a species and then putting them into the testicles of mice to see how they react. If normally there's no serious interaction. However, if you inject the rats with testosterone, if you inject them with estrogen, this estrogen is going to cause a feminization of the. It's going to interfere with the normal androgens that are within the rat's body, including the testicular cells. And when you do that, then the inflammatory response of the injection of the foreign tissue material is activated. So estrogen in this particular case is causing the activation of the immune cells in an area that is normally immunoprivileged or is normally suppressing the immune system, right? So, well, that's interesting. So this would indicate, this would probably mean that if you are a man and you are expressing more estrogen than you normally would. So, for instance, if you have a lot of fat cells, those fat cells will help convert testosterone into estrogen through aromatase. If you are producing a lot of estrogen, this might be influencing your autoimmune diseases, and it might be influencing the ability for your own immune cells to attack the tissues within your testicles. But I would also argue that the tight junctions, this is where the robustness comes in. So even after that happens, you probably still have your tight junctions are probably playing a very large role in still protecting your developing germ cells from your immune system, even if this immune privilege has been lost through estrogen production, right? So, but it would leave you more vulnerable, right? So if you are producing more estrogen, then you would want to avoid having your balls be inflamed, because then you lose your tight junctions functioning properly, right? So then if you have both estrogen and blue balls, then you could be seriously affecting your fertility by allowing your immune system to not only be activated within your testes, but then allow the white blood cells to secrete antibodies or move into this lumen, the lumen of the seminiferous tubules, right? So just something to consider as a consequence, I should say. These studies indicate that local testosterone concentrations characteristic of the testes seem to play an important role in the maintenance of testicular immune privilege. So remember, since the testosterone is being produced in the testes, it is still going to have a proportionally higher concentration of testosterone than the other parts of your body. So even if you are producing estrogen in your fat cells or in other locations of your body, it might not be enough to overrule the testosterone that is concentrated in your testicles. However, the precise manner in which testosterone mediates its anti-inflammatory functions on testicular leukocytes is as yet unknown. What can be surmised from the available data is that the conventional model of testosterone action through a nuclear receptor is extremely unlikely, as no evidence of androgen receptor expression has been found in testicular immune cells or in mature peripheral T and B lymphocytes. So what they're saying here is um, for testosterone for any hormone really, for any hormone to exhibit an effect on a cell, that cell must have some receptor or some way to understand that hormone. And what we're saying here is that the immune cells that are in the testes are localized in the testes. If you run experiments on what types of proteins they're expressing, they're not expressing a receptor to testosterone. So this tells us that whatever effect testosterone has on this immunoprivilege, it is not from it directly interacting with the white blood cells, with the immune cells. It has something to do with it interacting with the tissues, and then those tissues are presumably creating some chemicals that are signaling to the white blood cells to not be activated or to suppress themselves in those areas. Intriguingly, testosterone has been shown to elicit a calcium influx in splenic mature T cells and macrophages, presumably through non-genomic surface receptors. Although a recent report found androgen receptor expression in CD4 positive splenic T lymphocytes by highly selective reverse transcriptase polymerase chain reaction, and thus did not completely rule out direct effects through nuclear androgen receptors. So let me make sure I read this right. So as I said, you can test to see what proteins are being expressed by a cell. And one of the ways to do that is to do a reverse transcriptase PCR, which is when you look at the RNA of the cell. So remember, the cell has DNA, the DNA gets transcribed into RNA, and the RNA gets translated into your final proteins, right? So if you do what is referred to as reverse transcriptase PCR, which is the same type of method that you can use to detect viruses, the RNA viruses. If you do that with the individual cells, you can see what proteins the cell is at least trying to translate or trying to make, right? So they found that androgen receptor expression is happening in the CD4 positive splenic T lymphocytes. So there is these white blood cells that are located in the spleen. They are they are expressing some receptor for testosterone in contrast to the immune cells that were in the testicles. So there probably is, we would presume that there is a direct androgen response that is causing a change in these cells, in the spleen explicitly. It appears likely that androgens exert their immunosuppressive function on testicular leukocytes either through non-genomic pathway or indirectly by regulating the balance of pro and inflammatory cytokine expression in the Sertoli, Leydig, and pertubular cells. So what I'm going to do is I will stop there and just chat for a bit. So the role of leukocytes, that's where I ended. So what do I want to talk about? So these are some of the strategies. Okay, so these are some of the strategies that your testes are employing in order to suppress the immune system in those locations, right? and it's good to know a lot of the background information about the structure, the physical structure of the testes, to understand how this is happening. So we therefore understand that it is a combination of, or we're presuming that it's a combination of the physical aspects of the tight junctions of the blood testis barrier in conjunction with the immunosuppressive cytokines and the immunosuppressive hormones that are being produced in the testes in order to protect it, right? Now, this is where there is an issue when it comes. Oh, okay. So sorry, the clip stops, so it probably automatically stops at 2 hours. So there is an issue when it comes to if say a virus gets into these cells, um, and unfortunately, for instance, with the SARS-CoV-2 virus, the SARS-CoV-2 virus utilizes the um, ACE2 receptor to get into cells, right? And you're unfortunately, the testicular tissue actually has some of the highest expression, RNA expression of the ACE2 receptor at all. This could play a role in why men experience worse pathology due to contracting SARS-CoV-2. That's one of the possibilities, right? With um, so here's an issue I need to present. If you, someone who has testicles, gets contracts SARS-CoV-2 or any virus that utilizes the ACE2 receptor, and you end up getting some orchitis, you end up getting inflammation of the testicles, which help allow the virus to get into those cells. Then, and this has been described for other coronaviruses, where sometimes if given the opportunity, if you give the coronavirus to model organisms, or if you give SARS or MERS to some people, orchitis has been seen. So inflammation of the testicles has been seen. If it is possible that we start uh, detecting the RNA from the virus in the semen, this presents a big issue in that there could be a long-term chronic damage to the testicles associated with that pathogenic infection that is very difficult to get rid of because the immune system isn't working there. Remember, the immune system is not there to a degree. So if the virus is able to get in there, it can persist there for long periods of time, right? So that presents so the long-term damage, that's a problem, and then also the long-term transmissibility is also an issue. So you might have been able to have fought off the virus completely, and you are thriving, right? You are living your best life. But if you happen to have the culture of the virus within your testicles and within your semen, it is still possible that even though your immune system has fought it off throughout your body, it hasn't fought it off in the testicles because the immune system is being suppressed there. And if that happens, you might be fine and you might test negative. And you might isolate for 2 weeks after you've tested negative, right? If it's still in your semen, though, you could theoretically infect someone else. And then the process starts all over again in the new victim, right? In the new host. So this is just something that I wanted to discuss because I remember I was watching, what was it? It was the JAMA Network, the Journal of American Medical. Well, it's gone right now, but it was the JAMA Network, which is a highly esteemed journal. And they do podcasts every once in a while talking about different papers. And they had one about a month ago where they talked about the detection of viral RNA, the SARS-CoV-2 RNA in the semen. And that's what would the word be? They joked about it a little bit because they said, why does it matter, or something along those lines, where, well, if you're having sex with someone, obviously you're going to be exchanging respiratory droplets, so it wouldn't matter, it functionally wouldn't matter whether or not you contracted the virus from breathing in those respiratory droplets or bioaerosols, or getting it from semen. Like, obviously, if you're having sex with someone, you're having such intimate, close contact. I mean, you're not socially distancing, right? So they laughed about it, but I don't think they fully considered the immunoprivilege aspect of what happens if the disease is present in the semen for long periods of time. So if people were much more monogamous, if we lived in a much more monogamous society, then both partners would probably catch a disease, and it would stay within that unit, and then it wouldn't continue to spread. But that is not the society that we live in. We live in a lot more. We live in a semi-promiscuous society, and therefore it would still be possible that you're with someone, you could break up, or you could be polyamorous, what have you, and you're both infected and you're both recovered, but it's still possible that you are a carrier and could theoretically find a new partner and infect them as well. So, I mean, this is something, even if your respiratory droplets are pure, right? Even if your lung material has been rid of the virus for significant periods of time, if it's there, what are you going to do, right? So this is just something that needs to be considered and talked about, and perhaps in a few months to a year, this might be something that a conversation that people need to have before they end up hooking up with someone. So practicing safe sex might also involve uh, having a discussion about your COVID history, right? So that could be, might not be what happens, but it could be in the future that that is what happens. So if someone is, say, taking care of a loved one who is very immunocompromised, for instance, they might want to consider not having sexual partners that have been COVID positive in the past, right? Unless there might be some sort of semen test that people do to verify that their semen is clean as well. I mean, I'm not advocating for it. I'm just reading the situation. This might be a future direction that people go in their dating lives. Okay. Anyways, with that, let me. Okay, so good. So the camera still records more than 2 hours. So with that, leave any questions down in the comments, and I'll try to get to them. And yeah, hopefully I'll be able to finish this by tomorrow. But things have been pretty hectic recently, so I don't know if the headspace is really the best to be doing this. Granted, it is a good distraction because you don't talk back. So, anyways, you have a good one, and I'll see you next time.